So in this study, we used phage immune precipitation sequencing, which is a novel, low-cost, and multiplexed antibody profiling platform that we developed. What makes it powerful is that it allows us to look at IgG responses in a completely unbiased way. So we're looking at hundreds of thousands of peptides from thousands of allergenic, viral, toxin, and human peptides. Um, our lab tends to study IgG from this lens. We use PhIP-Seq to capture the entire landscape of IgG responses. It gives us a broad picture of what someone has been exposed to in their environment and how their immune system has responded. So their past and ongoing immune exposures. And what we found through this study is that we can use PhIP-Seq to identify IgG patterns that are distinct between allergic and non-allergic children, and they're also distinct in early childhood versus at birth. We observed that a greater proportion of peanut allergic children had IgG responses to peanut peptides, and this was similar for the egg allergic children in that they had increased IgG reactivity to egg peptides. This was during childhood and not at birth. I think that there are several possible explanations. This might reflect repeated dietary exposure, ongoing immune recognition, or maybe the fact that IgG and IgE may share shared epitopes. And we also know that B cells can class switch, so IgG can be converted into IgE, but mechanistically they're like very different. PhIP-Seq isn't intended to diagnose food allergy, and elevated IgG shouldn't be interpreted as protective or diagnostic. And we think of IgG more as like a marker of past and ongoing immune exposure, and it's not like a clinical test. So cord blood IgG reflects antibodies transferred from the mother across the placenta during pregnancy. And the IgG that we measure in a child's plasma reflects their own immune responses. And this is shaped by like their diet, their infections, and environment. And since we didn't see differences between peanut or egg differences at birth, this suggests that IgG patterns may develop after birth, so postnatally, and early childhood might be a critical window where the immune system is maturing and environmental exposures may shape these antibody responses. Yeah, so this was really interesting that we found this, that children who later developed milk allergy had lower IgG reactivity to milk peptides in cord blood. So this raises the possibility that milk-specific IgG could be protective, but PhIP-Seq captures total IgG and it doesn't differentiate between different subclasses, and different subclasses have different functions. So it's really important, and there could be future studies in the future that look more closely at effects that are different between the subclasses. Yeah, they're consistent with that idea, but we can't really conclude that from this study. The finding shows that that maternally transferred IgG may play a role in shaping the immune system early on, but this is still associated within our study, and there needs to be more mechanistic studies, such as profiling different IgG subclasses to test that directly. So this was also a pretty interesting study. We don't think that this represents molecular mimicry between wheat and other allergens, so there's probably not a true cross-reactivity. And instead, this may reflect broader immune dysregulation. It could be related to epithelial barrier dysfunction or genetic variation in HLA that influences how IgG recognizes different wheat antigens, and wheat IgG might be a marker of altered immune exposure. So like changes in how and changes in how someone responds to wheat, and it's not really a specific wheat allergy. Yes, that's the possibility that if gut permeability is increased, then more dietary antigens can enter the systemic circulation, and this can generate broader IgG responses. We didn't directly measure gut permeability in the study, so that would be a really interesting next step.